Hi, yeah, I'm Allison.
Hi.
Yeah. And yeah, we'll be presenting our rare plant ramp research 2023 to 2024. But first, we're going to talk about the ramp program that we're a part of. Um, so, a little bit about rare plant ramp. RAMP stands for Research and Mentorship for Post-Baccalaureates, specifically in biological sciences. It's an NSF funded program in multiple disciplines, but the San Diego Garden applied for this program with 3 other collaborating botanic gardens in 2021. And Alli and I are part of the first year cohort. There'll be 2 more years of this, so 2 more sets of mentees coming in next year and then again the year after. But the objectives of the program is to support networks that provide full-time research, mentoring, and training for recent graduates who lacked biological research or training in undergraduate studies in college. And so, yeah, Alli and I are fortunate enough to have this position. It's been amazing. We're 11 months in and excited to show you guys the research that we've been doing. But as I said, there's multiple ramp programs, but the rare plant ramp specifically for the Botanic Gardens is focused on conservation, plant science, and botany.
Yeah, so pretty much how it works is there are 4 gardens across the US with 2 mentees each, all of each garden containing also mentors to help the mentees start their research and help them guide them through it throughout. So our mentees from the garden are Rachel Stowski, Ben Naiman, Colin Corey, Todd Michael from the Salk Institute, and Ari Novi. And these this is us at Atlanta Botanic Garden in the beginning of the year when we all first met each other.
Yeah. So I'm going to start first on my project, focusing on Phaseolus filiformis, also known as the Slim Jim bean, comparing the salt tolerance of this crop pod relative to the cultivated and wild Phaseolus vulgaris, which is the common bean. And so I want to start off the background of this project on the importance of salt tolerance and why I want to look into this. So the motivation of this is soils are getting saltier. And irrigated agriculture land where people are using fertilizers. Fertilizers are just salts, and plants take them up, but also they get left behind in the soil. And if there's not enough irrigation to leach them through, then they accumulate in that root zone, and that can be really detrimental to plants, especially salt-sensitive plants like Phaseolus vulgaris, which is a glycophyte, also meaning sweet-loving plant. Basically, it's very sensitive to soil. And so these 2 graphs, the one on the top is the production of Phaseolus vulgaris globally, and underneath, there's a lot going on, but what I want you to focus on are the dots, which are soil samples. Green is not salty, red is extremely salty. And so there is some overlap, and there's a need to protect salt-sensitive plants like Phaseolus vulgaris from soil salinity, and a way of doing that is finding genetic resources in close wild relatives for potential breeding a more salt tolerant bean. So that's the premise of salt tolerance and why it's important. But I'm going to go into the common bean, Phaseolus vulgaris, first. It's in the Fabaceae, the legume family. It's an extremely agriculturally significant species. It's the second most socially and economic important leguminous crop. Does anyone have a guess to what the most important legume is? Yeah, soy. Soy, yeah, it's pretty obvious. Can't make soy sauce out of pinto beans, but still super significant. It's native to Mesoamerica regions of Mexico and Guatemala. Its wild habitat is dry forest, and that's kind of a nuance of all across Latin America. And it's been domesticated twice in Central America and in Argentina, and it's been domesticated for over 8,000 years. There's an estimated 40,000 varieties, so that's a lot, but you might know the pinto bean or the black bean, kidney bean, white bean, green bean, schoolmaster's goose bean, beefy resilient Grex bean, and 39 whatever more, 39,000 more. But yeah, it's a super diverse species. And as I said, it's extremely agriculturally significant. It feeds hundreds of millions of people. It's extremely high in nutrients and protein and fiber. 28 million metric tons are produced worldwide, And yeah, it's a stable food crop for hundreds of millions, meaning that hundreds of millions of people fully rely on beans as to get most of their nutrients. And just for reference, as 2 times the protein, 5 times the fiber, 4 times the iron than corn-based counterparts. Here's different varieties and their nutrient contents. It's, yeah, very diverse, even though it's one species. And another interesting thing that I like to note, it's a part of the 3 sister crop system, which is an indigenous agricultural practice of the Iroquois and Cherokee. If you've heard about this before, it's basically a holistic agricultural practice of the corn stalk used as a trellis for beans to grow and fix nitrogen for the squash and the corn that the squash acts as a ground cover. And it's just kind of a really nice agricultural system that has incorporated beans for, I think, 7,000 years. Again, just really pumping how important beans are. It's a very global food. These are just examples of some countries' dishes of beans, but you know, not most, but a lot of the world has been raised on rice and beans, as have I, and many of you probably, or I don't know. But here are just some examples of how people have eaten beans globally. Very diverse, very global. I'm just trying to say the common bean is important. Let's move on. So the other bean, Phaseolus filiformis, the Slim Jim bean. So why am I looking at this plant? It's really closely related to vulgaris. Here's the phylogeny. I know it's really hard to see, but the pink here is vulgaris and the green is filiformis. They're not direct sister, or they're not extremely closely linked, but they're sister clades. So they're still very similar. They have really similar genomes and could still be used as a genetic resource. But they look a little bit different. They're much vinier, leaves are smaller, flowers are smaller. The seeds actually look a lot more like gravel, sand-like. And their habitat is desert washes. So I actually took this picture in Baja, and this desert wash, it probably gets to 115 degrees in the summer, but this is in the fall, just starting to sprout. But definitely a different habitat and growth pattern. And so if you've heard of this bean before, it's probably because you know about it from the garden. It's known to be rare in California, but first I'm going to talk about how it's not actually rare in most of its range. It's pretty common in Baja California, northern Mexico, and the rest of the southwest of the United States. Not California. California is a different story. But this is my first time seeing Phaseolus filiformis. It was before I had started growing it, or it was my first time seeing it. It was in Baja, and it grows everywhere. Everyone thought it was silly that I was so excited about it, but in my head it was rare, and I was excited. But yeah, so it is rare in California. It was actually rediscovered by our SciCon team after not being seen for 40 years in the fall of 2022. This is a picture of Colin seeing it. Surprised, I wasn't there, but I heard it was very exciting. And we actually had the opportunity to go back this spring and found this massive population that they hadn't found the last time they went scouting for it, or however many times they went scouting for it. This massive mat, which I don't know if you can tell the size, but it basically covered this flat area, and it doesn't, the way I've seen it, it's not grown this big. And so that was really exciting, really close to this California occurrence. And the fact that there's this one rare occurrence in California, while it's rare or very common everywhere else, that in its range is its own question. It's like 100 miles away from its closest occurrence. So that's confusing, but I don't really address that. So someone else should, though. But so why am I looking at the common bean versus the Slim Jim bean? Common bean, super agriculturally important, huge staple food crop. We should look on how to make it salt tolerant. Slim Jim bean, as I said, the phylogeny, they're very closely related, and their habitat's pretty interesting that they're able to live in these super sandy desert washes. They're probably drought tolerant, so maybe they're salt tolerant. And then there's also been a lot of papers that have looked at the whole Phaseolus genus, trying to test. Finding salt tolerance in crops is a very normal thing to research, and people have done it in Phaseolus, and there's been hints that filiformis could be the most salt tolerant based off of a few papers that have looked at filiformis. But the thing with these papers, they only really look at the early life stage. They never go to fruit. So it's hard to say if a plant is salt tolerant when you just look at its baby version. It could be salt tolerant as a baby plant, but not produce any fruit. So that's kind of what I set out to do. To answer my three main questions I had for this project. First, is filiformis actually more salt tolerant than vulgaris, the common bean? And then I'll go into my range of seeds I have, but is there variation of salt tolerance across the entire range of filiformis, including California, but across its whole range, is there some occurrences that are more salt tolerant than others? And then if there is variation, can we identify correlations between why some are more salt tolerant than others to environmental conditions? For example, are more salt tolerant accessions coming from drier climates or higher elevation or less precipitation, things like that. So going into the methods, I'm going to start off with talking about the seeds because I think that'll make things make more sense moving on. So just like at a botanic garden, you accession a living tree that's in the conservatory, and on that accession it says where the plant came from and all this other data. You can get these accessions of seeds from these seed banks that come with where the seeds were collected or the original seeds were collected, with its GPS coordinates, who collected it, the year, and then they're usually seed bulk, so it's not exactly the same. But basically, you can order these accessions from seed banks like the CIOT and USDA, which is where I ordered these seeds from, or anyway. And these seeds come with GPS coordinates, which allow you to look at more those relationships between environment and variables that you're looking at growth. But I ordered 35 accessions of Phaseolus filiformis, 5 accessions of wild Phaseolus vulgaris, and you can see the beans are a lot smaller than the cultivated, but I also ordered 5 accessions of cultivated Phaseolus vulgaris, which is the agriculturally used vulgaris. So talking about filiformis specifically, in the pink is the range, and then the flowers are the coordinates of where the seeds that I ordered originated from. So from these seed banks, this is basically all of the filiformis you could order. This is kind of the best you can get. So we tried to capture the entire range of filiformis for this project. And this is basically, yeah, the best that you could get. But yeah, these are where the seeds come from. And so the experimental design, I did a greenhouse trial with a randomized block design. If you happen to see this, this is up in the medicinal greenhouse at the garden. How I attempted to do it was 3 replicates, but they weren't true replicates because I couldn't grow everything at the same time. There's just too many plants. But for the sake of simplicity, we're going to call them replicates, even though they're kind of spaced out by a month. But within each replicate, there's 2 treatments. The control, which was not treated with any salt treatment, and then the salt treatment. So when I irrigated them with sodium chloride and water versus just water. Within each treatment, there is the 35 accessions of Phaseolus filiformis, 5 accessions of wild vulgaris, and 5 cultivated vulgaris. So from the time of the first leaf expansion is when I started the salt treatment, and then they grew for 95 days. Throughout those 95 days, I collected a bunch of continuous data. At the end of 95 days, I harvested them, measured the biomass, the above ground biomass, and the total yield produced by each plant, and kept it all labeled, obviously, and to analyze later. And I took a bunch of data with this, but we're only going to look at yield for this. To answer these questions of is filiformis more salt tolerant than vulgaris? Question 1. First, I'm going to talk about what is salt tolerance, because it's actually more complicated to think about than you'd assume, or at least when you're actually trying to measure it, you have to think about how you want to measure it. There's a lot of ways to measure it, but the overall premise of salt tolerance is that the plant will not respond worse in salt treatment than in a control treatment. For example, say filiformis here, it produces and grows the same way under control treatment and a salt treatment, versus vulgaris, it might grow really well under a controlled environment, but once it's treated with salt, it completely dies or does really bad. So in this instance, filiformis is salt tolerant, vulgaris is not salt tolerant. So change of fitness in salty environment, less change, more salt tolerant. Regardless of how productive it actually is, it's relative to how it's doing in the control. And so as I was saying, I'm only going to talk about yield in this, but at the end of each trial, I collected all the seeds, weighed them out, and yield is obviously the most agriculturally significant. If you're thinking about breeding, you want whatever you're trying to produce, or whatever salt tolerant plant you're trying to produce the most seed. That's the end goal. So looking at seed produced, and these are just examples, these are the accessions. For this accession, the no salt treatment produced 2.07 grams of seed versus the salt treated produced 2.22 grams. And so the salt tolerance yield is 2.22 divided by 2.07, 1. So the closer to 1, the more similar the salt and not salt had reacted, versus less than 1 salted worse, more than 1 salted better. So the result of is filiformis more salt tolerant than vulgaris? This is actually a pretty simple answer here. On this graph is the survival rate of by each species. So here filiformis, the controlled, about 90% of the control survive versus around 75% of the salt treated survive. Then we get to wild vulgaris, about 80% vulgaris under the control survive, but then only 1 out of the 15 wild vulgaris survived, and the same goes for cultivated. So basically, based off of just the survival rate, you can see that vulgaris isn't very salt tolerant. It dies. And then I tried showing that even more through calculating the salt tolerance, but there wasn't even enough vulgaris under the salt treatment to calculate salt tolerance. So to answer the question, filiformis is more salt tolerant than vulgaris. There's definitely statistical significance in there about just around their survival and lack of producing anything. But then we're going to go into the more difficult to answer questions of, is there variation of salt tolerance across the range of filiformis? So now we're kind of leaving vulgaris. We now know that filiformis is more salt tolerant, but is there a difference across the range of its salt tolerance? Are there more accessions that are more salt tolerant than others? So I tried to answer this question. So this graph here, all of these are different accessions of filiformis and their relative salt tolerance based off of that calculation of the previous slide. So as I said, I had 3 replicates and they were spaced out by time. So there's a lot of variation in how well these replicates did. But saying that, there's definitely a spectrum of salt tolerance across the range. Now, this actual statistical significance between each accession, I mean, we can't necessarily say which ones are the most salt tolerant or which ones are the least salt tolerant, but we can kind of see by this, these ones are more salt tolerant than these ones. And so having this kind of spectrum, while we can't say anything's too exact quite yet, we can still ask that question of, is there any correlation between the spectrum of salt tolerance and environmental variables like climate or precipitation, elevation, things like that. And so to answer this question, is there variation correlated with environmental variables? The first thing I did was mapped it out. So here are all the accessions again, but they're labeled by their salt tolerance index. And if you look closely, it might be hard to see. It's very random, the salt tolerance index is. So there's a 1.6 here, a 2.7 right next to a 1.9, a 0.38 right next to a 1.4. So at least when you're looking at proximity to each other, it's hard to see if there's an area where there's a higher salt tolerance. And this is actually a lot of area, so that doesn't necessarily mean much. We can look still at environmental variables, but yeah, these are the top 5 accessions from that previous bar graph, and they're all over the place. It's not like there's one region that has really salt tolerant. It seems kind of random just by looking at this map, but we still went to go look at the variation correlated with environmental variables by pulling data from these basically databases that have WorldClim has these 19 bioclimate variables. some being like average temperature, annual precipitation, but then also environmental stresses like the hottest temperature and the coldest temperature, the wettest month, the driest month, um, as well as variability between seasons. And you can pull this data as well as I pulled terrain data from the USGIS to get elevation. And from elevation, you can get a bunch of different things like aspect and slope and things like that. And basically try to correlate, see if there's any correlation between these salt tolerance values and these different variables. And so it's just a bunch of data manipulation and running models. And with that, there's no significant relationship between these bioclimatic variables, elevation, location. There's a slight relationship between salt tolerance and average temperature, but the p-value is really insignificant, or not really insignificant, but it's still not a threshold of that's significant. But to be said, there's a lot of variation in my trials, so there could be further explanation if there's a more robust experimental design. But there's a big chance that it's likely random, this salt tolerance, but could use further investigation. But in conclusion, filiformis is more salt tolerant than vulgaris. That's pretty obvious just because vulgaris can't survive in salt conditions. And that there does appear to be salt tolerance variation within filiformis. However, there's too much noise in the data to say exactly an order of the most salt tolerant accessions. And there doesn't appear to be a relationship between salt tolerance and environment, at least looking at yield. There's other things that are in the works to be looked at, but future work, if I were to do this again, I would do a more robust experimental design, more replicates so there could be less variability, and maybe focus on less accessions instead of the whole range, focusing more on the most salt tolerant ones to really see if there's actually no correlation between salt tolerance and environmental variables. And then I'm currently looking at physiological responses that might explain salt tolerance, so less about which is more salt tolerant, but more explaining why things are salt tolerant, like the water and salt accumulation in leaves, increase or decrease in leaf mass per area, or increase or decrease in the number of leaves grown over time. And then lastly, which is something I'm really excited about that I just have recently worked on, is they're currently working on the genome assembly of Phaseolus filiformis. So we just got this graph yesterday, but I've been working with the Salk Institute and Todd Michael's lab doing the whole genome assembly of filiformis, the California accession, the rare one. And we just assembled it all. So this figure is all the contigs that will be assembled or is assembled into the whole genome that we looked at now. And it's currently running through some supercomputer to annotate for all the genes that correlate, or trying to find all the genes it can, I guess. It's machine learning. I just learned about it yesterday, but really excited about it. And then hopefully the goal is to be able to compare that genome assembly and those genes of interest that are specifically correlated to abiotic stress tolerance to the Phaseolus vulgaris whole genome. That's the idea, but that's in the works also. And thank you to everyone who has helped this project happen. It's been awesome. Everyone at the San Diego Botanic Garden and Salk Institute. And yes, thank you, everyone. It's been great 11 months. I'm going to take it away to Alli.
Hello. We're moving to Eriodictyon. So what is Yerba, which is Eriodictyon? Well, it's a genus in the Boraginaceae family, or the borage forget-me-not family. It goes by the common name Yerba, which translates in Spanish to holy herb. And this is because the Spanish came over and saw the indigenous tribes using this plant for various ways to, or for various uses, such as for respiratory disorders, as topical treatments for wounds. So I was kind of given this umbrella term as holy herb. And species in this genus have funnel to urn shaped flowers ranging from white, lavender to purple with leaves that are colline and alternate. This is one example. This is Eriodictyon crassifolium. So I believe that there's three really unique things about this genus that I would like to point out. One being that it's a genus that's very limited in its range. It pretty much only grows in California and nowhere else in the world. As you can see, so this is all the species in the genus, and for the most part you see that sometimes bleed into Oregon, sometimes into Baja, but for the most part it's just California, which is pretty crazy, an entire genus in one state. Along with that, second unique point about this genus is that it's only 10 species, which is a really small genus. And 4 out of those 10 species are either federally or state listed, meaning that 40% of this genus is considered a rare species. The third unique thing about this genus is the research that has been done on it. So it's mainly been looked at for its healing abilities against oxidative stress, its anti-inflammatory activities, and also discovery of novel treatments for Alzheimer's disease. And a woman named Pamela Mayer from the Salk Institute has looked more closely into this for mainly Eriodictyon californicum, which I'll get more into, but also for other species of this genus for its abilities to protect cells from cell death and flavonoids that it's producing. So what is a flavonoid? Well, flavonoid is a compound or molecules that are produced by plants for immune responses to help it cope with certain stressors such as UV radiation, predators, etc. So it's very beneficial for the plant, but back in these papers, we're kind of trying to understand how it might be beneficial for humans as well. And so what Pamela Mayer from the Salk Institute found was that 3 flavonoids, strubin, Eriodictyon, Homo-Eriodictyalis, seems to have a strong correlation, at least in Eriodictyon californicum, with its abilities to protect cells from dying, which is what patients with Alzheimer's disease experience. They experience neurons in the brain dying. So as I'm reading these papers, I'm looking at these tables. On your left-hand side, we're looking at a table of Eriodictyon californicum that's highlighted and its levels of strubin. And on your right-hand side, we're looking at a table of Eriodictyon traskii and its levels of strubin. I have a bias with Eriodictyon traskii because I worked on Catalina Island before I came here, and Eriodictyon traskii is a plant that grows on Catalina. So my eye automatically goes here. But so to go back to Eriodictyon californicum and the strubin levels, it has a very wide range of strubin that's produced. It's shown to have the highest levels of strubin produced to not even being detected. And so when we look at Eriodictyon traskii and its strubin levels, it's showing that it's also not detected. But if we look even closer, we see that n equals 1, meaning that they only looked at one plant. And it was an herbarium specimen from like the mid-1900s. If we looked at Eriodictyon californicum, they sampled 82. Like, that's not fair. Another point to look at is its Eriodictyon traskii. We're not really certain if it's Eriodictyon traskii subspecies smithii, which is the subspecies that grows on the mainland near Santa Barbara, or if it's subspecies traskii, which is the plant that grows on Catalina and nowhere else in the world. So I was like, this needs more research, in my opinion. Thus, a project is born. I started a multivariate phytochemical analysis of Eriodictyon traskii, subspecies traskii, a Catalina Island endemic. And I had 3 main questions. First off, does Eriodictyon traskii actually contain strubin, or is the table right and have no detection at all of strubin? Also, going back to what we see with the wide variation of strubin being produced, why is there such a big variation? Is there something that's influencing these strubin levels? So I wanted to know, could it be something with the season? Is it being produced during certain times of the year? Is it because of the leaf stages? If it's a mature plant or a mature leaf, does some pathways in that leaf cause the strubin to be produced? We don't know, but I want to find out. Thus, we take off to Catalina. And so the methods for this is, since we want to know if seasons play a role in the variation of sterubin production, we have gone so far 3 times to Catalina to 4 separate populations. And so we've gone in the fall, we've gone in the winter and the spring so far, with summer yet to come later this month. Each time that we go to the same populations and collect from the same exact plants. These 4 separate populations was Death Canyon, which is down a very steep ridge. 2 was White's Exclosure, which is a population enclosed by a fence built by the Conservancy to help prevent disturbances from deer and bison. 3 is Middle Fire, which is a population that was burned in 2016, and Eriodictyon decided to be the first to take over that area. So it is the most abundant and healthiest population that we collected from out of the 4, versus 4, which is Cape Canyon, which is the most disturbed population we collected from. It was a roadside population where the facilities goes and collects their dirt. So it's constantly just having people dig at it and tractors come and whatnot. And for the most part, all of these populations were pretty easy to get to, except for Death Canyon in that steep way. But we all had our own ways to get down there. Ben had his own way. I walked down. So, what did we do when we got to these populations? Well, we got to the plants that we collected from. We made sure to tag them, note them on ArcGIS. That way we could find them again. And we collected both mature leaves and small leaves from those plants that were then taken to the lab, made into extracts. So all those good molecules, flavonoids we want to look at, were taken out of those leaves, dried down, and re-diluted with DMSO into these little extracts that we could then take to the lab to conduct phoroptosis assays. Phoroptosis is kind of just a fancy word for cell death. And so I actually had the opportunity to go to the Salk Institute and work with Pam Mayer, which is the lady that worked at the Salk Institute and did the big research on Eriodictyon californicum. And I was able to mimic her methodology that she did for Eriodictyon californicum on Eriodictyon traskii. It was awesome. It felt like I was meeting someone famous. And so pretty much we grew HT22 cells in the lab and put them in 96 well plates, exposed them to 3 different threats. And what I mean by threats is 3 different compounds that are found in pathways of Alzheimer's disease that are known to be the causes of cell death of those neurons that the patients experience. So we have the cells, we have the 3 different threats, and then we add our samples to it, and we see if the samples pretty much block the threats from killing the cells. Along with that, we were able to do some liquid chromatography mass spectrometry, which I know is a mouthful, but pretty much it is just using a really fancy, expensive machine to tell us what molecules are in those extracts. So the phoroptosis assay results that I did at the Salk with Pam pretty much showed that in the fall, the collections of the younger leaves had less protective abilities compared to the mature leaves. However, once we start looking at the winter and spring, those young leaves start to come up back and meet the mature leaves. So I have a hypothesis that maybe, because when we collected those fall collections, it was October, it was hot, there was no water, it was dry, the plants aren't growing during that time. So my hypothesis is that those young leaves aren't producing all of those really good molecules that the plants need until it starts hitting the growing season, which is winter and spring, once it finally gets water and is like, okay, time to grow. But it's just a hypothesis. So then putting that with the LCMS work that we're looking at, I'm going to try and go through this as best as I can. So these 5 lines that we're looking at, or let's look at A through D. A through D is all from the same plant. A and C is total ion chromatogram, which is just meaning that it's just a big bowl of everything. And then when we look at B and D, we're looking at extracted chromatogram. We're looking at strobin, Eriodictyon, homoerydiktyon, those target flavonoids that we're really interested in. So we're going to focus on B and D right now. B is the young middle fire leaf that was collected in the fall that I have circled over there. So it's that purple bar in the red circle. The mature middle fire leaf, and then we're looking at the mature middle fire leaf, which is that blue bar that's in the red circle. And E is a strobin standard. So that's pretty much telling us where we can find that flavonoid strobin on these graphs. And if we look closely at this, you can see D. That's pretty much the exact same peak, right? So what does that tell us? That means that there's strobin in there. And if we look at B, we're looking, you could still see a little peak of where that strobin is, but just not as much. And if we take these two and we put it back to there, you can see that that purple bar with this is smaller. The blue bar is bigger. So there's a good correlation. So better protection abilities correlates with more strobin, possibly. This is kind of preliminary data. We still have a lot of analyzing to do, but this is a very exciting first start. So kind of conclude with this big information that we have, all this information that we have. All collected Eriodictyon tissue exhibits some properties that protect against cell death. So in one way or another, if we look at the controlled versus the actual extracts, there is some sort of protection for all tissue, whether it's 60%, 80%, 90%, all tissues seem to have some level of protection. There is also a possibility of, or the possible correlation between strobin levels and protective abilities, as we just saw. The variation in cell protection properties and fluctuation of flavonoid levels across all seasons is still under analysis. So you remember how that was my second question? Still a big question for us, but if we go back to this graph, all these red bars are my control, and we see that there's a lot of fluctuation in it. It's because I conducted all these ferroptosis assays at different times, and HT22 cells, the cells that we used in this study, there's a lot of variability with them. Say they've been left in the freezer for a longer period of time, they were grown different way, they're gonna die either better or not as well compared to that. So I'm planning on collecting my summer tissues and then retesting them all at the same time to make it a very controlled experiment. That way we can actually look between all seasons and answer that second question. And then lastly, as I was talking about before, this research is aimed to understand flavonoid protection and their abilities to combat neurodegenerative diseases such as Alzheimer's. And this is pretty important because, you know, in the lab, it's easy to tell, like, there's a correlation between strobin and protection, but Botanic Gardens is a very nice way to get a deeper look at these things, because without going into the field and doing this type of research, you won't know, or it's harder to tell when you want to collect it. Like, say this goes to clinical studies and we want to be able to harvest this plant when it's at its peak strobin production. We won't know that unless we do this type of work. So, yeah. Oh, sorry, did you think that was all? Oh no, there's a part two. Oh, you really did. No. I also was like, you know what? Let's take this a step further. Let's look at germination of Eriodictyon. And like I said before, Eriodictyon is a really small genus, so that's why that's why there was this opportunity to do widespread germination trial in the entire genus. Well, 8 out of the 10 species. I was able to get my hands on 2 seeds of these species, Eriodictyon altissimum and Eriodictyon tomentosum. Eriodictyon altissimum specifically, it is a 1b2 plant, meaning that it's rare in California and everywhere else. So she's really rare. She was really hard to get seeds for. So I was like, yeah, in tomentosum, it was just, we tried looking around, but it was just hard to, you can't win them all. But my idea for this germination trial came from the Santa Barbara Botanic Garden germination trial on Eriodictyon capitatum, which is, oh, sorry, Altissimum is a 1B1. Eriodictyon capitatum is 1B2. And Santa Barbara Botanic Garden did a germination trial on Eriodictyon capitatum, and they found that there was a significant increase in germination with the liquid smoke compared to dry heat and scarification. And not only that, but they also looked at the difference between a light setting and a dark setting. And they found that in the dark setting, not only did they have higher success rate with germination, but it also took less days to germinate. So I was like, I'll take that as a basis. And I created my own germination trial for these 8 out of 10 species. And I used 5 different groups. I used a control light. As you can see here, these are the Petri dishes. So they're given no treatment, they're exposed to light. Then we have a control group, which was no treatment, but covered in foil, so they're in a dark setting. And then we have a liquid smoke treatment, which was seeds that were soaked in 10% liquid smoke solution overnight. A dry heat treatment, where I put them in the oven at 100 degrees Celsius for 5 minutes. And then a combination group, which was both the liquid smoke and the dry heat together, all covered in foil. And so what I found was that overall, between most of the species, there was pretty good success of the liquid smoke and the combination groups together. However, the dry heat treatment was not as successful. As you can see, it's the pink bar. Don't see a lot of pink, but you see a lot of yellow and blue, which is the liquid smoke and combination, which makes sense. Seeds are smart. Say like there's a fire that goes on. The seeds aren't going to be like, oh, fire, okay, let's go. They're going to probably wait for a little bit of rainfall to come. So I was like, okay, I get it, guys. And not only that was the success of germination better for the combination liquid smoke group, but also the average days that it took for the dry heat trial was significantly higher of the compared to the liquid smoke and the combination group. So overall, dry heat, not good treatment. Another interesting thing to note is that Eriodictyon perrii has a lot of argument on whether or not it should even be in this genus. A lot of taxonomy stuff. But it's interesting to see that it's the only species out of all the ones that were treated that had 0 germination across all of them. So I'm like, perrii, bye. And then, sorry. And then these graphs are more so looking at a time frame. So I checked these seeds every 3 days for 63 days. And, some took better than more. Like Angustifolium really loved the liquid smoke. Capitatum also really liked the liquid smoke. But Traskii seemed to really love the combination group. It had not as much success with the liquid smoke group, but for some reason it really preferred the combination group, which you can see a lot of different blues, but it's the dark, that one right there. So in conclusion for this, these results suggest a correlation between germination of Eriodictyon species in post-fire conditions, at least for most, not all. Liquid smoke in the combination trial had significantly higher success rates compared to the dry heat in the controlled groups. Like I said before, 0 germination for perrii. Something else to know is that, so I had 2 different treatments. The second treatment I had used plant preservative mixture because in the first one, they got so moldy. The agar plates, as I'm sure some of you know, mold grows on agar. And it made it really hard to identify which seeds had germinated. So I actually reached out to Heather Snyder, who did the Eriodictyon capitatum germination trials. And I was like, what do I do? And she was like, use ppm. So the second trial was a lot more successful than the first trial. So if I had a chance to redo this, I would want to go back and redo that first trial with ppm. And I think for future studies, it would be really interesting to look at gibberellic acid along with the liquid smoke and combination trial. Gibberellic acid is a compound in seeds that are used to help make seeds germinate. So I feel like that could also be interesting to add into the trials. Also, all seeds that have germinated are given a chance at life and uppotted. As you can see, these are some of my Eriodictyon seeds that we have germinated. They're going, and we plan on outplanting them and maybe using them for future research. Yeah. So I'd really like to give a thank you to everyone at the garden who's helped me make this possible. Literally could not have done it without them. Also outside organizations, some of y'all are here. But literally, like, can't express it enough, like how awesome everyone is. And also I'd like to thank all of you for coming and listening. Any questions?